Hello, warm welcome to the lecture 6th of cell biology. In this lecture we are going to see the next chapter, that is the nucleus. Myself Dr. Sunil Patil from Loknitya Venkataraw Panchavati College, Nasik. In this lecture we are going to see first the syllabus of the nucleus, the points to be discussed here, that in detail. In nucleus we are going to see the first point, that is shape, size, number and position, then ultrastructure of the nuclear membrane and pore complex. Then third point we are going to see the nucleolus, general organization of nucleolus, chemical composition of nucleolus and functions of nucleolus. Then fourth point we are going to see is the nuclear matrix or nuclear sap, that is nucleoplasm. Then fifth point, that is the nucleocytoplasmic inheritance interactions, nucleocytoplasmic interaction. This point we are going to see in the next lecture, that is lecture 7th, as a separate part, although it is from the chapter nucleus. Then we are starting the general characteristic feature of the nucleus, a large membrane bound, it is important word, membrane bound, usually spherical protoplasmic structure within a living cell containing a cell hereditary material, that is, and contributing to metabolism, growth and reproduction. Cell hereditary material, that is chromatin or chromosome, and it is double membrane structure. So it is important feature of the nucleus. Then other characteristic feature, nucleus as a cell organelle. It was first discovered or described by Robert Brown in 1831. Structure of nucleus of eukaryotic cell become distinct in a non-dividing cell, means not in cell division or during the interphase. In interphase we can study the structure of nucleus in detail, or well we can see the structure of nucleus, each and every part. In eukaryotic cell, in prokaryotic the nuclear membrane is not there around the DNA or the single circular DNA, so that is not called referred as the nucleus, that is called as the nucleoid instead of nucleus. So they are the cell without nucleus, prokaryotic cell. Eukaryotic cell, eu means true, karyon means nucleus, where true nucleus is there, so they are called the eukaryotic cell due to nucleus, the name is there. Such an interphase nucleus is made up of nuclear envelope, nucleoplasm, nucleolus and chromatin network. So we are going to see these parts in detail in this lecture: nuclear envelope, nucleoplasm, nucleolus and chromatin network. Each and every part of these four parts in detail. Some sieve tube of vascular plant cells and erythrocytes of mammals, that is RBCs of mammals. These two types of cell lacks the nucleus. There is no nuclei in these cells, sieve tube plant cell and RBCs of mammals. RBCs of mammal and sieve tube of plant cell does not have any nucleus in the cells. So these are some characteristic features of the nucleus. So you can see the double membrane bond structure, then the nuclear pores, nucleolus, then same things are there. We are going to see the structure in detail. First we are going to see the shape of nucleus. It depends on the shape of cell, internal environment of cell and the physiological stage of cell. Generally it is spherical, but may be elliptical, oval or irregular in shape. The shape is generally spherical or round, but we are going to see the different type of cell. For example, epithelial cell having disc shaped nuclei, then paramecium having kidney shaped nuclei. These are disc shaped nuclei in the epithelial cell. This is kidney shape or horseshoe shape macronucleus. In the paramecium, muscle cell having columnar type of nuclei. You can see the elongated or columnar type of tall nucleus. So it is the muscle cell. In WBCs they are irregular in shape. You can see it is bilobe, it is multilobe in neutrophil, then in monocyte with a notch, in lymphocyte it is round but with a notch, it covers the entire part. So in leukocyte it is irregular in shape. The second point is the size of nucleus. It depends upon the volume of cell, amount of DNA, amount of protein and metabolic state of the cell or number of chromosomes. The size varies from 3 to 25 micrometer in diameter. So we have studied the shape and size of the nucleus. Then nucleoplasmic ratio is the next point. There is a definite ratio between size of nucleus and volume of cytoplasm. So size depends on the volume of cytoplasm. It is also called as nucleoplasmic ratio or nucleoplasmic index. Nucleus of haploid cell is smaller always than the diploid cell of the same cell. Nucleoplasmic ratio can be determined by volume of nucleus divided by volume of cytoplasm minus volume of nucleus. So nucleoplasmic ratio, volume of nucleus, and VC equal to volume of cytoplasm. So this formula is important in view of examination. It is always asked for define or explain what is nucleoplasmic ratio. You have to write the formula and this NP equal to nucleoplasmic ratio, Vn equal to volume of nucleus, Vc equal to volume of cytoplasm. Volume of nucleus divided by difference between volume of cytoplasm and nucleus. It is the nucleoplasmic ratio. Then we are moving to the number of nuclei in a cell, nucleus in a cell. Generally one nucleus is present in a cell. However, there are certain eukaryotic cells which shows wide variation in number of nucleus. On the basis number of nucleus, the cells are following types. A nucleated cell means cell means not the cells without nuclei, they are anucleated cell. The cells without nucleus, example erythrocytes of mammals means RBCs of mammals, sieve tube of higher plant. So these are the RBCs which are without nucleus, where WBCs are with nucleus in the blood of mammals. Then second thing is uninucleated. Most cells are uninucleated, means one single cell with single nuclei. Usually many plant and animal cells are uninucleated. Muscle cell, nerve cell, WBC, these are uninucleated cells. Then binucleated cell is the next point. So we have already seen the paramecium with two nuclei. This is called as the macronucleus, this is called as the micronucleus. Mega or macronucleus, it is large kidney shape, and small round is the micronucleus. So binucleated cell paramecium is prime example. Other is the liver cell, cartilage cells, etc. Multinucleated cell, the cells with three or more nuclei, they are called the multinucleated cell. More than two nuclei, they are called the multinucleated cell. Example is the osteoblast cell of the bone marrow, which form the bone cells. Then striated muscle fibers. You can see the single muscle fiber having many nuclei. The single cell having many nuclei, it is called the multinucleated cell. So osteoblast cell and muscle fiber, striated muscle fiber only, not smooth. Smooth having single nucleus. These are multinucleated cell. So anucleated, uninucleated, binucleated, and multinucleated. According to the cells are referred by the number of nuclei in them. So we are going to see the characteristic feature or structure of nucleus in detail. So first we are starting from the covering of nucleus, that is nuclear envelope. The nucleus is enclosed by double layered cellular membrane, means two membranes around the nucleus, they are called the nuclear envelope. The two membranes are separated from each other by the space 10 to 30 nanometer. The outer membrane ONM is connected to the network of tubules and sacs, that is called the endoplasmic reticulum. Outer membrane, it is connected to this network of endoplasmic reticulum. The outer membrane also showing the ribosomes attached to it. So outer membrane is rough, while inner membrane is connected with the fibrous protein, so it is smooth. So outer membrane is rough and inner membrane is smooth. The space between the two layers is known as peri-nuclear space. It is fluid-filled space. The nuclear envelope separates the content of the nucleus from the cytoplasm. The outer membrane is rough, we have already seen, due to ribosome. The inner membrane is smooth as it is connected with the fibrous protein called as the nuclear lamina. So this is first point, that is the nuclear envelope. Then we are moving on the second point, that is nuclear pores. The number of membrane is, the nuclear membrane is pierced with holes. That holes are referred as the nuclear pores. These pores allow the nucleus to communicate with the rest of the cell, as they control the passage of substances into the cell, into the nucleus and out of the nucleus. The number of nuclear pores are correlated with the transcriptional activity of the cell. The cell with more transcriptional activity have more nuclear pores. The nuclear pores 50 to 80 nanometer in diameter and octagonal in shape. These are enclosed by circular structure called as the annuli. Annulus is singular, annuli is plural. So actually 2 annuli per pore is present. The 2 rings or annuli, they are interconnected with spikes. The pores or annuli together called the pore complex. It consists 2 annuli rings with inside diameter of 80 nanometer. 8 spikes are extended from the rings. Human cell having 3000 to 4000 nuclear pores. So these are the 2 rings or annuli. These 2 annuli are connected with the spikes. This is nuclear pore complex. This is the outer membrane. This is the inner membrane. So a ring is present to the outer membrane, one is present to the inner membrane. They are connected with the 8 spikes. 4 are seen in the figure of one side. So we are going to see the nuclear pore complex in detail. So same things. This is first ring. It is entirely a ring, annulus. This is another ring, another annulus. They are connected with the spikes. So it can close and open the pore. Nuclear structural nubs are there. The nubs, fragmental nubs and structural nubs, they can close and open the pore with the help of the spikes and annuli. So this is again same thing. Then we are moving on the next part, that is third part of the nucleus, that is nucleoplasm or karyolymph. Plasm means fluid present in the nucleus, that is nucleoplasm. Karyon means nucleus, lymph means fluid, same wording. The nucleus contains nuclear sac or semi-fluid matrix enclosed by the nuclear envelope. This is known as the nucleoplasm or karyoplasm. It holds the nucleolus and the suspended chromosome or chromatin. It is composed of nucleoprotein, proteins, enzymes and minerals in the nucleoplasm. It consists chromatin. You can see the network of thread, it is called the chromatin. This is nucleolus, and this part where they are floating, it is the nucleoplasm or karyoplasm. These are nuclear pores, 3 number. Then 2 number, they are ribosome, which are present on the outer surface and on the endoplasmic reticulum. The 4th is nucleolus, 5th is chromatin fibers. The relaxed extended state in the interface of the cell, if they are chromatin, they become chromosome when compact. This is the outer membrane of the cell. So this is nucleoplasm or karyoplasm. The nucleolus is the 4th part. Inside the nucleus, nucleolus is present. You can see this is the nucleus, where 1 number, it is the nucleolus. Nucleolus or central part of nucleus has dark dense spherical area known as nucleolus. The content of nucleolus is continuous with the rest of the nucleoplasm, as it is not membrane bound structure, means there are no clear demarcation between nucleolus and nucleoplasm, as it continues. Only the dense part of the karyoplasm or nucleoplasm called as the nucleolus. The nucleolus is the production factory of ribosome, which is the in turn manufactured proteins. Ribosome later produces the protein. Thus it can be said that the nucleolus is indirectly involved in protein synthesis and RNA synthesis. It consists DNA of nuclear region. 4 types of rRNA are produced. Ribosomes are produced and RNA binding proteins are produced. Depending on the synthetic activity of a cell, there are one or more nucleoli present. Nucleoli may be 1 or more. It depends on the synthetic activity of the cell, and size of nucleolus may be different. For example, cells of oocyte contain large nucleolus, where the sperm cell contain small incomplete nucleolus. The nucleolus takes up around 25% of the volume of nucleus. About 25% is the nucleus of the total volume of nucleus. Then we are going to see the chemical composition of nucleolus. Nucleolus is usually divided into three parts: fibrous, granular and matrix. It is region found within the cell nucleus that it is concerned with the production and assembling of the ribosomes. So it is prime function of the nucleolus, production of ribosome. The nucleolus is composed of RNA and protein which form around specific chromosomal region. The main composition of the nucleolus are protein, RNA and traces of DNA. This structure is made up of ribonucleic acid, RNA. Its main function is rewrite ribosomal RNA and combine with the protein. It is the function of nucleolus. It then sends the subunit out of the nucleus and combine into the complete ribosome. There is a uninterrupted chain between the nucleoplasm and interior part of the nucleolus, as it is continuous part, nucleolus and interior part of the nucleoplasm, which occurs through a system of nucleolar passage. Nucleolus is usually divided into three parts, namely fibrous, granular and matrix. We have already seen as the first point. This is granular part, pink color dots, that is granular compounds. Then some fibrous compounds are there in the nucleolus and dense fibrillar components are there. So fibrous, granular and matrix are the parts of nucleolus in the composition. So this is the nucleolus. It is the region found within the cell nucleus that is concerned with the protein assembling and cell ribosome. It is rewritten of the function, same thing. Then we are moving on the functions of nucleus as a separate part, as it is asked for the short part. First point, that is ribosome, how the ribosomes are formed in the nucleolus. It is important in the view of examination. The nucleolus is ribosome producing site. Ribosomal RNA genes are there of the nucleolar organizer region, transcribed by RNA polymerase first and rRNA. RNA polymerase first and rRNA of the nucleolus transcribe the genes into ribosome. Ribosomal proteins are synthesized in cytoplasm. These proteins are synthesized out of the nucleus, and these are entered in the nucleus through the nuclear pores or nuclear envelope. The rRNA is packaged with that ribosomal proteins which comes from cytoplasm. So rRNA and ribosomal protein combine to form ribosomes. In this way, 45S ribosomes are manufactured in the nucleus. During this 45S ribosome lose some of its RNA. Then it gets split into separate precursors. Large and small subunits are formed. We can see the 45 is divided into 18S and 25S, or in this way. The precursors are 18S, 28S, 5S, 8S, etc. rRNA are produced. These subunits are now transported outside the nucleus, and they join in the cytoplasm, join to form the ribosome. In this way, ribosomes are produced. It is as per short note, so it is important part. Then second function of the nucleolus is formation of rRNA. Actually it is first, then ribosomes are produced, but as short note is asked, so we are taking that first one. So nucleolus, by the process of transcription, involving formation of various kinds of rRNA which required for the subunit of ribosome. Then third is the protein synthesis. Nucleolus is involved in the production of subunit which then together form ribosome. Therefore nucleolus play an important role in protein synthesis and production of ribosome. Maggio in 1960 and other scientists have suggested that the protein synthesis takes place in the nucleolus. Some of the biologists suggest that ribosomal proteins are also synthesized in the nucleolus. The fourth function is the role in mitosis. Nucleolus play important role in the mitosis, as it also acts as the nucleolar reorganizer. So these are the functions of nucleolus. Then we are going to see the chromatin network, which is present inside the nucleus. In the interface, we can see as a chromatin. The interface nucleus has highly extended and elaborate nucleoprotein fibers in the form of chromatin. They are called as the chromatin fibers. Then DNA gets associated with protein molecule to form chromatin. DNA plus the histone protein mainly called as a chromatin. During different stages of the cell division, cell shows structured chromosomes. In cell division, they become compact. You can see the chromosome. Highly compact, the compact nature you can see. Each loop having the 8 histones, means it is octamer, called as the octamer, and it is DNA fiber is coiled around. This is the DNA fiber only, and these are the histone proteins, and there are also non-histone proteins. Chromatin contain DNA and some basic proteins called as the histone, some non-histone protein as well as DNA togetherly form the chromatin. A single human cell has approximately 2-meter long thread of DNA distributed among 46 chromosomes, means all chromosomes may form the 2-meter long DNA. So it is highly coiled in the cell. The chromosomes are made up of heterochromatin and euchromatin. So we have to see what is heterochromatin, what is euchromatin. Heterochromatin is the region which remains highly condensed during the interphase and is typically non-transcribed. So heterochromatin and euchromatin are the two parts of the, it may be part of the same chromosome, the active and inactive state. The part which remains uncondensed is called as the euchromatin. Uncondensed means it can transcribe, and which can be transcribed. Heterochromatin is composed of 25 angstrom fibril thickness, where the euchromatin is the 30 to 80 angstrom thickness. So euchromatin are more thick than the heterochromatin. Heterochromatin is considered to be genetically inert. It cannot transcribe, so it is genetically inert. As it is not involved in synthesis of DNA, so heterochromatin are genetically inert one. Euchromatin are active part of the chromosome. So chromosome may divide according to its activity. The function of nucleus, togetherly, nucleus is the core of cell and it performs various vital activities. It is responsible for protein synthesis, cell division, growth and differentiation. The first function is metabolism. Nucleus control all the vital activities of the cell. It is a regulatory organelle of the cell. It regulates all its activities through the specific proteins. Heredity is the second function, an important function, since the nucleus contains genetic material, that is chromatin or chromosome, in the form of DNA molecule in the chromosome. The genetic material transfers the characters from parents to offspring or one generation to the next. The Hammerlin's grafting experiment. It is asked for the 2 marks short question. Hammerlin grafting experiment with the single cell green algae acetabularia show that nucleus of a cell contains genetic information that directs cellular development, which directs the cellular development. So next functions of the nucleus. Differentiation is the third one. Nucleus controls all the cell differentiation during the embryonic development, during the development of particular cell into specific tissue or specific cell, such as nervous tissue, epithelial tissue, connective tissue, etc., or muscular tissue. They are called the specific tissue. Such development is controlled by the nucleus through gene expression. Then fourth function is the RNA synthesis. The synthesis of ribosomal and other RNA synthesis takes place in the nucleus. Fifth function, exchange of material. Nuclear membrane is concerned with the exchange of material between cytoplasm and that nucleus. And then nucleus gives support. Nuclear membrane provides surface attachment with the cytoskeleton, that is microtubule and microfilament. So it gives support to the shape size of the cell. Then genetic code. Nucleus contains the master plan for the protein synthesis in the form of triplet codon. So these are functions of nucleus. 7 functions of nucleus you have to remember. Then one part is the nucleocytoplasmic interaction. We are going to see in the lecture 7th. That is the next lecture. So see you again. The next lecture is nucleocytoplasmic interactions. So thank you very much. If you have any doubt or problem, you may ask me directly. So this is about the nucleus, but next one from the same chapter is remaining, that is nucleocytoplasmic interactions. So thank you very much.